Hello friends, warm welcome in this video, that is the C4 pathway. Now we will see what is this C4 pathway. Many plants either do not photorespire at all or they do so to only a limited extent. These plants have a normal rubiscose and their lack of photorespiration is a consequence of mechanism that concentrate CO2 in the rubisco environment and thereby suppress the oxygenation reaction. Three mechanisms are suggested for concentrating CO2 at the site of a carbonylation. These three mechanisms are: first, C4 photosynthetic carbon fixation, that is a C4 pathway; second, resolution acid metabolism, that is CAM pathway; and third, CO2 pumps at the plasma membrane, which is particularly occurs in algae and cyanobacteria. So now we will learn in detail the C4 photosynthetic carbon fixation or C4 pathway. Early labeling of C4 acids was first observed in 14CO2 labeling studies of sugarcane by H.P. Kostrak and colleagues and of maize by Y. Karpilov and co-workers. When leaves were exposed for a few seconds to 14CO2 in the light, 70 to 80% of label was found in the C4 acid malate and aspartate, a pattern very different from the one observed in the leaves that photosynthesize solely via the Calvin cycle. In pursuing these initial observations, EMD Hatch and C.R. Slack elucidated what is now known as the C4 photosynthetic carbon cycle, that is the C4 cycle. They established that the C4 acids, malate and aspartate, are the first stable detectable intermediates of photosynthesis in leaves of sugarcane, and that carbon atom 4 of malate subsequently becomes carbon atom 1 of 3-phosphoglycerate. The primary carboxylation in these leaves is catalyzed not by rubisco but by phosphoenol pyruvate carboxylase. The manner in which carbon is transferred from carbon atom 4 of a malate to carbon atom 1 of 3-phosphoglycerate became clear when the involvement of mesophyll and bundle cis cells was elucidated. The participating enzymes occur in one of the two cell types. PEP carboxylase and pyruvate orthophosphate dikinase are restricted to mesophyll cells. The decarboxylase and the enzymes of the complete Calvin cycle are confined to the bundle cis cells. When with this knowledge, Hatch and Slack were able to formulate the basic model of this cycle. So when we study C4 cycle, there are differences in leaf anatomy between plants that have a C4 carbon cycle and those that photosynthesize solely via the Calvin photosynthetic cycle, that is the C3 plants. A cross section of a typical C3 leaf reveals one major cell type that has chloroplast, the mesophyll. In contrast, a typical C4 leaf has two distinct chloroplast containing cells types, mesophyll and bundle sheath. This is called as kranz anatomy. There is a considerable anatomic variation in the arrangement of the bundle sheath cells with respect to the mesophyll and vascular tissue. In all cases, however, operation of the C4 cycle requires cooperative effort of both cell types. No mesophyll cell of a C4 plant is more than 2 or 3 cells away from the nearest bundle sheath cells. In addition, an extensive network of plasmodesmata connect mesophyll and bundle sheath cells, thus providing a pathway for the flow of metabolics between these two cell types. This is the tissue which contains only mesophyll cells, whereas this is the tissue in which the bundle sheath cells are covered with the mesophyll cells. So only this C4 plants contains the bundle sheath cells with a chloroplast, whereas these C3 plants lack this bundle sheath cells. This is called as kranz anatomy. So the basic C4 cycle consists of 4 stages. First is fixation of CO2 by the carboxylation of phosphoenol pyruvate in the mesophyll cells to form a C4 acid, malate and or aspartate. Second, transport of C4 acid to the bundle sheath cells. Third, decarboxylation of the C4 acids within the bundle sheath cells and a generation of CO2 which is then reduced to carbohydrate via Calvin cycle. Fourth, transport of C3 acid, pyruvate or alanine, that is formed by the decarboxylation step back to the mesophyll cells and regeneration of a CO2 acceptor phosphoenol pyruvate. So this is the basic structure in which the various steps are shown. This is the carboxylation, this is the regeneration, this is the decarboxylation. So these are the steps which are occur in C4 cycle. So in this way, the communication between the two types of cells takes place. This is the phosphoenol pyruvate, which is in the presence of a PEP carboxylase and bicarbonate converts into oxaloacetate. This bicarbonate is formed in the by the uptake of atmospheric CO2 by carbonic anhydrase. This is converted into bicarbonate. Now this oxaloacetate in the presence of malate dehydrogenase and NADP, HET converts into malate. So this malate enters into the bundle cyst cells, and here this is converted into pyruvate in the presence of a malic enzyme. Again here is a requirement of a NADP. So here the CO2 is removed, and this CO2 enters into the Calvin cycle. Again this pyruvate enters into the mesophyll cells, and this pyruvate converts into phosphoenol pyruvate by the enzyme pyruvate phosphate dikinase. So in this way this reaction is taking place. One interesting feature of this cycle is that regeneration of the primary acceptor phosphoenol pyruvate consumes two high energy phosphate bonds, one in the reaction catalyzed by the pyruvate orthophosphate dikinase and another in the conversion of pyrophosphate to 2 orthophosphate catalyzed by pyrophosphatase. Shuttling of metabolites between mesophyll and bundle cyst cells is driven by diffusion gradients along numerous plasmodesmata, and transport within the cells is regulated by concentration gradient and operation of specialized translocators at the chloroplast envelope. The cycle thus effectively shuttles CO2 from the atmosphere into the bundle cyst cells. This transport process generates a much higher concentration of CO2 in the bundle cyst cells than would occur in equilibrium with the external atmosphere. This elevated concentration of CO2 at the carboxylation site of Rubisco results in suppression of the oxygenation of ribulose 1,5 bisphosphate and hence of photorespiration. This pathway is discovered in the tropical grasses, sugarcane, maize, etc. The C4 cycle is now known to occur in 16 families of both monocotyledons and dicotyledons, and it is particularly prominent in graminaceae, which involves corns, millets, sorghum, sugarcane, and chenopodaece, as well as it occurs in cyperaceae. About 1% of all known species have C4 metabolism. There are 3 variations of the basic C4 pathway that occurs in different species. The variations differ principally in the C4 acid, malate or aspartate, transported into the bundle cyst cells and in the manner of a decarboxylation. The net effect of a C4 cycle is to convert a dilute solution of CO2 in the mesophyll cells into a concentrated CO2 solution cells of a bundle cyst. Studies of a phosphoenol pyruvate carboxylase deficient mutant of Amaractus edulis clearly showed that the lack of an effective mechanism for concentrating CO2 in the bundle markedly enhances photorespiration in a C4 plant. Because of higher energy demand, C4 plant photosynthesize under non-respiratory conditions, that is high CO2 and low O2 conditions, require more quanta of light per CO2 than C3 leaves do. In normal air, the quantum requirement of a C3 plant changes with the factors that affect the balance between photosynthesis and photorespiration, such as temperature. By contrast, owing to the mechanism built in of high photorespiration, the quantum requirements of a C4 plants remain relatively constant under different environmental conditions. Light regulates the activity of key C4 enzymes. Light is essential for the operation of the C4 cycle because it regulates several specific enzymes. For example, the activities of PEP carboxylase, NADP malate dehydrogenase, and pyruvate orthophosphate dikinase are regulated in response to variation in photon flux density by 2 different processes: reduction oxidation of ethyl groups and phosphorylation dephosphorylation event. NADP malate dehydrogenase is regulated via the thyrodoxine system of the chloroplast. The enzyme is reduced, that is activated, upon illumination of leaves and is oxidized, that is inactivated, upon darkening. PEP carboxylase is activated by light dependent phosphorylation dephosphorylation mechanism yet to be characterized. The third regulatory member of the C4 pathway, pyruvate orthophosphate dikinase, is rapidly inactivated by an unusual ADP dependent phosphorylation of the enzyme when the photon flux density drops. Activation is accomplished by the phosphorylatic cleavage of this phosphate group. Both reaction, phosphorylation and dephosphorylation, appears to be catalyzed by a single regulatory protein. In hot, dry climate, the C4 cycle reduces photorespiration and water loss. Two features of the C4 cycle in C4 plants overcome the deleterious effects of higher temperature on photosynthesis that were noted earlier. First, the affinity of PP carboxylase for the substrate bicarbonate is sufficiently high that the enzyme is saturated by bicarbonate in equilibrium with air levels of CO2. Furthermore, because the substrate is bicarbonate, oxygen is not a competitor in the reaction. This high activity of PEP carboxylase enables C4 plants to reduce the stomatal aperture and thereby conserve water while fixing CO2 at rates equal to or greater than those of C3 plants. The second beneficial feature is that suppression of photorespiration resulting from the concentration of CO2 bundles its cells. These features enable C4 plants to photosynthesize more efficiently at higher temperatures than C3 plants, and they are probably the reason for the relatively abundance of C4 plants in drier, hotter climates. Depending on their natural environment, some plants show properties intermediates between the strictly C3 and C4 species. Thank you.